Have you been afraid of cooking meat because you may not be confident the quality of cooked meat will be as juicy, tender, and chewy as expensive steak? Meat tenderness is the most difficult to predict, which related to the strength of the structural components of the muscle fiber such as the myofibrillar mass, sarcoplasmic proteins, and connective tissue that is evaluated by instrumental method and sensory. Meat tenderness is a quality parameter affecting the tough muscle and changes into soft meat. Meat tenderness depends on different factors such as post-mortem pH, type of tissue, and temperature. In cooking, tenderizing is a process to break down collagens in meat to make it more palatable for consumption. There are 3 different methods for tenderizing meat, which are mechanical, marinating, and chemical. Mechanical tenderization involves breaking down the meat's connective tissue with sharp blades. Marinating a steak in acids or enzymes break down the fibers and tenderizes the steak. Next, food-grade phosphate are used in meat products for several reasons such as changing and/or stabilizing of pH value, increasing water holding capacity in order to higher yields, decreasing losses of weight in cooking, improving texture and sensory properties. The objectives of this experiment are to study meat tenderization by applying different treatments, to compare and measure the effectiveness of each treatment. In order to making the comparison of tenderization of meat, materials that we need are meats, pineapple, phosphate, and blade. Firstly, the raw meat is cut into suitable pieces and weighed. Then the meat sample is divided into 4 samples to indicate different tenderization process. The raw meat then go to the observation of color. Next, preparation of pineapple is done to making puree. The pineapple is blended without addition of water. Then, the puree is weighed for 50 grams. 3 grams of phosphate is dissolved into 100 milliliters of water. Next, the raw meat is put in the Ziploc bag. Pineapple puree and phosphate solution is filled in the Ziploc bag containing raw meat to immersed. For marinating purpose, samples that immersed with pineapple puree and phosphate solution is transferred into the lab scale tumbler. The opening of the tumbler is checked to make sure it is closed tightly. Then, the tumbler is set rotation for 20 minutes. While waiting for tumbling process, mechanical method was applied by using meat tenderizer hammer. After 20 minutes, the marinated meat is removed from the tumbler. The meat samples is rinsed with tap water to remove the excess puree on the surface of meat. Next, all of meat sample will be analyzed to determine the color, water holding capacity, and shear force. To observed water holding capacity, the meat is cut into 3 grams of each sample, while for shear force analysis, the meat is cut into 3 centimeters cubed. Determination of water holding capacity is by load the 2.5 kilograms of load on the meat samples. Next, determination of tenderness of meat sample is done by using Warner-Bratzler shear force. After that, the remaining meat samples is put into the water bath at 80 degrees Celsius for 30 minutes. After 30 minutes, the samples is removed from the water bath for analysis process. The excess water is removed from the meat sample by dapping with tissue towel paper. Next, the meat is cut same as shear force analysis of raw meat samples. The observation of cooked meat samples shear force of is done with triplicate. Lastly, the data obtained is recorded and saved. Based on the result for shear force test, the uncooked meat with phosphate showed the highest value, while the cooked meat with enzyme had the lowest value. The lowest value of shear force indicated higher value of meat tenderness. Enzyme increased the reaction rate as the bromelain in pineapple helped to break down connective tissue and produce flavorful meat. Next, water holding capacity WUC for uncooked meat treated with enzyme showed the highest value than the meat treated with phosphate and mechanical treatment, as the reaction of enzyme helped meat to hold larger amount of water and increase the meat quality. For the color determination, the lightness L value of uncooked meat treated with phosphate was the highest due to high in pH level. Next, the redness of value of uncooked meat applied with mechanical treatment was the highest as the technique did not interfere with the color changes in meat. The yellowness B value of uncooked meat treated with phosphate had the lowest value, while the uncooked meat treated with enzyme had the highest value. In conclusion, the most effective tenderization method was using enzyme followed by tumbling process compare with other methods, mechanical and injection of phosphate. It also results in high water holding capacity, which enhances the textural qualities such as tenderness and juiciness. Enzyme treatment is the most effective tenderization method as it is inexpensive and yields the lowest shear force value. Next, it results with high water holding capacity, which enhances the textural qualities in terms of tenderness and juiciness. The value of low-value tough meat cuts can be increased via marination with enzyme by transforming them into tender meat cuts. However, treatment with enzyme can cause an aftertaste flavor to the meat, which influence the sensory attributes to the cooked meat. It also contributes to highest value of a yellowness, which obviously comes from the pineapple itself that predominant by carotene. The appearance can be factor contributing to the consumer product acceptance, where bright red color of fresh was more preferable. The value of low-value tough meat cuts can be increased via method of tenderization by transforming them into tender meat cuts.